Well, thank you very much for the invitation to be here. And I always dread going after Paula, but I did include some of Paula's work in my talk as well. And as a little bit of a competition, Paula, I thought I'd try to go a little broader platform than you did. Yours was pretty broad. And so Paula talked about using a really amazing layer-by-layer work in nanoscience for different kinds of medicine. I thought I would talk today about using a platform, virus-based platform, to go from basically the beginning of Earth to solar to cancer. So what's the connection between all of those? And the work that I do here in the Koch Institute is very collaborative. I work with a lot of collaborators I'll talk about as I go along. And so, as promised, I've been really interested in how nature makes materials. Examples are biocomposite materials like this abalone shell that's 98% by mass calcium carbonate, 2% by mass protein, yet it's 3,000 times tougher than its geologic counterpart. All made in ocean temperatures, ocean pressure, doesn't use toxic materials, doesn't add toxic materials back into the environment. Wow, what a wonderful manufacturing idea. And every time a male and female abalone get together, they pass on the genetic information that says, this is how to build an exquisite material. Here's the DNA sequence, go out and build your own. And so they go out and build their own shell. Same with diatoms made out of silica, also made in the ocean. And so what my lab has been interested in doing is giving genetic information to non-living structures like solar cells and batteries so they can enjoy some of the same principles that biology does, which is self-assemble, room temperature synthesis of materials, correction, the ability to correct. And so when we think about future technologies, we start with the beginning of the Earth about 4.5 billion years ago. Only took about 1 billion years to have life on Earth. And then all of a sudden, really amazing things happen. You have multicellularity, you have photosynthesis. But it wasn't until about 500 million years ago, during the Cambrian geologic time period, I promised you I'd get all geologic time in here. During the Cambrian geologic time period, that organisms started making hard materials. Before this time, everything was soft and squishy. So why was it more difficult to make calcium carbonate or silica-based shells than to do photosynthesis? Well, the answer was that the oceans changed during this time. There's increased iron, increased calcium, increased silicon, and now organisms had the possibility to start expanding their toolkit for making materials. And so they did just that. And so they went on to make materials that would become bones, that would become glass made at room temperature, and would become calcium carbonate. And so what our goal was to do was to convince biology to work with the rest of the periodic table. It was very limited over this 500 million period of time. And so we use simple organisms like bacteria and viruses and yeast. Let's see and convince them to start making new kinds of materials, such as a virus-based battery. And so we work at the interface between nanotechnology and biomaterials. And we work on energy, healthcare, and the environment. And people always say, you know, Andrew, why don't you focus on just one thing? And I always say I do focus on just one thing. I focus on using biology to make better nanomaterials. So those nanomaterials have increased functions. And that can be in solar cells and batteries, fuel, enhanced oil recovery, CO2 sequestration and storage, more abundant materials, cleaner mining, water purifications, therapeutics, and imaging. And to me, they're all very closely related because by controlling the way atoms interact with each other, controlling where they are in a particular surface, it controls the morphology of that structure. It controls the optical, electronic, magnetic properties of that material. And so biology has already learned how to do that very well through 4 billion years of evolution. Let's borrow ideas from biology. And we work a lot with companies. And as mentioned, I founded 2 companies, an electronics company and a natural gas company based on this technology. Here's some natural biomaterials. Coccolithophore unicellular algae makes calcium carbonate. This is the abalone shell. If you take that abalone shell and you break it and you look at it in the scanning electron microscope, what you'll see is it's made out of these beautiful tablets of the metastable crystal structure of calcium carbonate, and that the thickness is perfect. It's defined by the particular specific kind of gastropod that makes it. And in between, there's an organic matrix. Same with diatoms. They're also genetically coded. Magnetotactic bacteria make small single-domain magnets, Fe3O4, Fe2O3. All of those are coded at the genetic level. But nature has done a pretty good job. It hasn't really worked with this part of the periodic table, except for we can talk about exceptions to that. or this part of the periodic table, or this part of the periodic table. And so our goal was to give the ability of biology to work with almost anything we wanted it to be able to work with. And so in order to do that, I said that organisms in the ocean about 500 million years ago started making materials. It took 50 million years to get good at it. So no one to this day has given me a 50 million year grant, or no students have signed up for a 50 million year PhD project. So we needed a more rapid way of doing it. Now what I didn't tell you is that the way that organisms learned how to do this in the ocean is they took proteins they already had that had a particular function, and they started modifying them. And so in the case of the abalone shell, what they have is they have these proteins that are very negatively charged, and they have these cells that secrete the proteins, and they grab onto calcium and build calcium carbonate based materials. And so the inorganic materials of calcium carbonate are bound to these proteins as nucleation points. And those proteins actually allow that inorganic reaction to happen, but also control the direction of its crystal growth by changing its physical optical properties, such like that. So in order to beat that 50 million years, we decided to use an idea from the drug discovery industry, which is phage display. I'm really a material scientist by training, so a phage to me is an object that's 880 nanometers in this direction and 6 nanometers in this direction. It has single-stranded DNA, and the single-stranded DNA codes for the proteins that make it up. But it's pretty easy. You can make a library out of this by taking the single-stranded DNA, cutting it to a random DNA insert, and now you can express a random peptide insert of interest on one end of the virus. And again, thinking of as an engineer, I look at this M13 bacteriophage. It's this absolutely amazing self-assembled structure. You can see the alpha helixes that make it up, and they self-assemble to make it up. Now imagine putting a DNA sequence in there that codes for a protein sequence, and now that's expressed, say, in 3,000 copies along the code of the virus. And now you can use that to start making materials or probing materials of interest. Because remember, the idea here is, oh, wow, there's all these elements on the periodic table. There's all these materials that are very interesting, but there's no genetic link between them because biology's never learned to work with them. How do you get those two ideas together? How do you get them to merge? That's the convergence. And so you take a library of like 1 billion possibilities and force them to interact with anything you want, because you're looking for that protein that will chemically and geometrically bind that material of interest. And ultimately what you want to do is not only bind, but you want it to be able to grow that material and then pass that ability off to its offspring, right? And so you go through and you screen, you look for 1 out of 1 billion that grows a better battery electrode. You put 1 billion in, most of them don't interact. You find some that interact. Viruses can't make copies of themselves. I'll use the word virus and phage. A phage is a virus that only infects bacteria. Bacteriophage, sorry. Bacteriophage only infects bacteria. You find one that recognizes it, and then you infect into its bacterial host. The bacteria makes 1 million copies, and now you ask it to bind again in more stringent conditions. You're looking at the 1 out of 1 billion that build a better battery. The 1 out of 1 billion, as Paula and I showed, that can build a better solar cell. And so, because these alpha helixes that are on this virus coat, they're crystallographically aligned, they self-assemble, and they're crystallographically aligned, you can think about them as a way of organizing, translating that crystallographic symmetry onto a material of interest, inorganic material, say. And so we've been able to do that, where we've been able to now to make these wire-like structures into, these are gold wires, and these are gold platinum wires, and Professor Schauhorn and my group together use these to make better fuel cells. We use these for ethanol fuel cells and methanol fuel cell applications, and showed that we can increase them up to 8 times. This is Paula and my students. Paula and I actually have a shared lab, and our students is what our students think we look like, and it's pretty accurate. And so Paula and I have been working on solar cells. How do you give genetic information to solar cells, batteries? I've worked in catalysis quite a bit. I have a company I founded in drop-in fuel replacements. How do you go from methane to ethylene, ethylene to polyethylene, ethylene into drop-in fuel replacements? We have a program in how do you go from waste CO2 into building supplies using yeast. And then we have a program in cancer diagnostics and imaging, which I'll tell you a little bit about today. And we have programs in artificial photosynthesis as well. But one of my favorite materials are carbon nanotubes. And most people know graphene better than carbon nanotubes. Graphene is basically a molecular sheet of carbon. And if you don't actually roll it, but you can imagine rolling it up into a tube, and this is a carbon nanotube. And carbon nanotubes are very small. They're nanometer diameters in this direction, and it can be very long in this direction. but they have a lot of interesting optical and electronic properties as well as mechanical properties. But they're a nanometer across. They're very difficult to think about how to pick up. But one of the projects we worked on, two very talented graduate students in my lab, Yoongjung and Hyungjung, they had an idea of what if we engineered viruses to pick up carbon nanotubes based on molecular recognition on their tips, and then we simultaneously engineered them to grow battery electrodes. This was iron phosphate, eventually lithium iron phosphate. And they were able to do that, and then they were able to get them to self-assemble, and they formed a record-breaking battery at the time that went to a lithium-ion battery that went to the White House, all based on molecular recognition. It had the gene to bind actually carbon nanotubes. Aha, I didn't talk about this because I'm not talking about batteries today. But to be a good battery electrode, either anode or cathode, you need to be a good ionic conductor and a good electrical conductor simultaneously. And that can be tricky because a lot of times those materials aren't the same. So their idea was to have the virus be a good electrical conductor, and then the rest of the virus to be a good ionic conductor to build this in self-assembled structures. Here it is. You had the M13 bacteriophage that grew the electrode material, and then you had basically a super highway to get electrons in and out of this material, and it actually self-assembled into the current collector. And this is the data at the time. This is specific energy versus specific power. This is a one-gene system. A virus has one gene. This is a two-gene system. This is our best two-gene system that actually was a really good battery at the time for specific power versus specific energy. And it was all based on genetically engineering these structures. Since then, we've done some really fun things. This is an aerogel foam battery that isn't published yet. It was with Professor Jin Kong's group here at MIT. That it's a very lightweight aerogel battery. It's a battery that's sitting on top of a little flower. That's a 99% porous structure. That's a lithium-oxygen battery electrode. Paula and I with our group, did the same idea of let's see if we can get engineer viruses to make inexpensive solar cells higher efficiency. And these are a kind of solar cell called dye-sensitized solar cell. And here's the idea in a dye-sensitized solar cell. You have titania nanoparticles. You have a dye, and you have an electrolyte. And the idea is you shine light, you promote an electron. That electron gets injected into a titania nanoparticle. It has to make its way through this titania nanoparticle network to a transparent conductor and out to the load to the alternate electrode where it's actually recycled. And our idea was, well, if we could engineer a virus to grab onto a carbon nanotube and coat itself with titania, maybe we can get a more direct electron path in a solar cell. So here's the idea how it would work without a virus. You shine light, you have an electron, it's making its way through, but all kinds of bad things can happen to it along the way. It can recombine with the dye molecule or the electrolyte and never make it. But if you instead have a virus that's in there that's binding these single-wall carbon nanotubes, you can shine light. It can find the carbon nanotube and get a direct path to the current collector. And when we did that, we increased 0.1% biology in that point, increased the efficiency by 33%. So a little biology can go a long way. And this is a paper recently with Seth Lloyd's group. He's a quantum a theorist here at MIT. We genetically engineered viruses to actually have this interplay between quantum and classical transport to increase exciton transport in light harvesting devices, which was super, super fun because it's engineering a virus to support quantum transport. One other non-cancer example was a company I founded called Siluria Technologies, which was the idea of how do you take methane, natural gas, and convert it from C1 to C2 chemistry. Now, people have worked on this for about 30 years in a way that would be low temperature. It's called low temperature oxidative coupling of methane. And the idea is that methane's very abundant. It's methane to ethylene, C1 to C2, and C2 is the most traded petrochemical in the world. And this is our factory in Houston that we opened last year that based on finding viruses to grow new materials, so those new materials could go from natural gas to ethylene, and we're making a ton of ethylene a day in this plant in Texas. So I was asked to work on cancer, and to me, again, it's going to be the answer is going to be nano, and the answer is going to be bio-nano because that's kind of what we work with. And so what we decided to do was put a team together between Mass General, MIT Lincoln Labs, which is an FFRDC, which Nan and Andy, two really fantastic engineers at Lincoln Labs helped to develop different prototypes for cancer imaging. I'll spend a few minutes telling you about that today. And so one of the things that we decided to look at was something called second window near-IR imaging. So imaging above 1,000 nanometers, or really between 950 and 650 nanometers in emission between 950 and 650 nanometers. And so what are the competing techniques? Well, there's MRI imaging, which has good resolution and good depth of penetration, but it's costly and long imaging time. There's CT, which has good penetration depth, but can be radioactive material for CT and PET scan. There's ultrasound, which has good resolution but poor penetration. And there's near-IR1, which is safe, but you don't get the depth of penetration that you can get with second window near-IR. But this requires sensitive equipment and a lot of new development. But our group decided to take on this as a way of looking to develop second window near-IR imaging for cancer. And the idea is that when you're imaging above 1,000 nanometers in the second window, that you could have a much deeper depth of penetration, and you would get signal-to-noise improvement about 100-fold. But there are not a lot of probes that are available here, or instrumentation that would be commercial available to carry this out. And when you think about imaging, you think about resolution versus of depth of field. And if you're looking at the micron depths, you're excluding scatter. If you're looking 1 to 3 millimeters, you're ignoring scatter. But if you're looking at the centimeter scale, you want to be able to embrace scatter. And so that's what we set about to do. We decided to first start by imaging and looking at ovarian cancer. So that's what I'm going to tell you about the rest of the time. The current gold standard in the clinic is contrast-enhanced CT for imaging ovarian tumors. And the sensitivity below a centimeter is poor, and it can be hard to resolve these due to low contrast. The way that ovarian cancer is treated right now is primarily through a process called surgical debulking, where once the patient is diagnosed, the surgeon goes in and removes everything he or she can see by eye and follows up with chemotherapy. And you can see from this metadata analysis that the survival rate that's measured in months is dependent on how well the patient is debulked. So this is suboptimal debulking, and this is optimal debulking. And so working with physicians at MGH, we decided to see if we could improve surgical debulking and find smaller tumors. And so what we started with was using M13 bacteriophage again, and screening in our library for the ability to pick up single-walled carbon nanotubes of a particular chirality and stack them along the length of the virus. Now we chose this because, one, we were good at carbon nanotubes, and two, they're fluorescent in this region of second window near-IR, and they have different fluorescent depending on which chirality of carbon nanotubes you choose. And so we built our own imager to begin with. We have a 2D indium gallium arsenide detector, an 808 laser, and we built a simple imaging system to be able to test our imager. What we did was we took an M13 bacteriophage that was carrying single-walled carbon nanotubes that were fluorescent in this region, and we engineered it on this end to bind to the protein SPARC, which is a protein that has been shown to be overexpressed in highly invasive ovarian cancer, and set about to test this in an animal model. Now, this is the mouse model that we looked at. This mouse model has xenograft tumors in the perineal cavity, and this is looking at near-IR2 with our imager. This is near-IR1, and this is visible. So I hope that you can see between near-IR1 and near-IR2, we can start to pull out tumors that we couldn't see in the near-IR1, or definitely not in the visible. And we also found that using the system, we had a 10-fold increase in the tumor homing as well. And this is looking at injection through the intraperitoneal space and then imaging after 24 hours. And if you remove these tumors, this is a photograph of what the tumor looks like, and this is the fluorescence that, from the carbon nanotubes. And this one is non-specific organs where we're not seeing fluorescence, and it corresponds with the H&E staining. And this is in the small tumors that were found on the liver. And so we decided to do an experiment where we did to test the debulking. And so in this particular panel here, you can see we don't have any background fluorescence. There's an animal there. We haven't injected with our probe yet. We inject with our probe, and this is an animal that has a high burden of ovarian tumors. And then we have a surgeon go in and do surgery without seeing this image, just do image by eye. And he removed everything he could see by eye and then sewed the animal back up, and he missed all these tumors. They were too small for him to see. We then had him go and look at this image and remove more tumors, and you can see that he could remove many more tumors once he had our map of where we thought the tumors were. And so in this, using this guided surgery, we were able to find tumors that were less than 1 millimeter in size when the surgeon had the ability to look at our map. And this is work from Paul and I that just came out in PNAS, where we're using the technology and the imaging system that we developed and combining it with her beautiful layer by layer technology to try to study the pharmacokinetics of the particles in the drug release in real time, because this is all real-time imaging. So we're pretty happy with that, but then our surgeon said to us, our collaborator said to us, what we really want to do is have a real-time surgical guided system, because the 5-year survival rate is less than 30%. What we want to do is in real time go in, find all the tumors, and test the survival with more optimal debulking. And so we set about to build a new imager. This was our 2nd generation imager. It's a real-time interoperative fluorescent detective system. It's a little more complicated than the last system. It actually has multiple lasers and different visible beam, as well as an IR in-band beam to be able to do the surgery and align where the tumors are and the fluorescents are at the same time. This is our team that put this effort together, and this is our real-time surgical guided mouse imager. And this is actually looking in-band reflectance and near IR2 fluorescence, and so this is with the skin closed, the skin open, and this is after the fluorescence guided resection. And then this is looking at the tumors we're pulling out in fluorescence versus the in-band reflectance. And so using algorithms developed by MIT Lincoln Labs, we're able to color code these in real time and now show the surgeons in real time. Here's all of the tumors we see between 1.5 and 2.5 millimeters now, between 0.5 and 1.5 millimeter. And so they can go in real time, find the very small tumors that are actually less than 1 millimeter in size, and do real-time surgical guided planning during the debulking process. And then we were able to develop software where we could go in and even in very small tumors, tell in those small tumors which parts we thought were actually were real tumors and which areas were tumor-free. And so far we have a specificity of 98%. So we went on to do survival studies, and in this example we have a non-guided surgery, and here we have our guided surgery. And this is 2 days 2 days pre-surgery. So we injected our probe into the intraperitoneal space, and here's 2 of the mouse patients. This is 10 days post-surgery. You can see that the surgeon missed a lot of tumors, and these tumors have regrown. You can see 10 days post-surgery here, there's no tumors visible to the eye. Now, normally a patient would get chemotherapy. Our mice didn't get chemotherapy here. At 3 weeks post-surgery, the animal has a very heavy burden of tumors, but we still miss some of the small tumors with even our guided system. At 5 weeks post-surgery, all the animals had died in the non-guided surgery, and you can see that, that we had quite a bit of tumor regrowth in our guided surgery. the experiments we're doing now is actually treating the animals with chemotherapy at this stage, hoping that we can get the smaller tumors. But our overall data shows an increase of 40% in survival with our guided surgery system versus our non-guided surgery system, which my collaborators told me is quite remarkable for ovarian cancer, because there's been so little progress in ovarian cancer over the years. We've then since gone and re-engineered our viruses to be very small. We call them inophage, which means small phage in Portuguese, and so that they could actually have better clearance and get deeper into the tumors. We invented a new way of using DNA origami to specifically cut our single-wall carbon nanotubes to very precise lengths. So now we have very small carbon nanotubes that bind to very small phage. And this is actually kind of a cool system. The DNA origami sets the spacing for how long the carbon nanotubes are. We go in and we cleave them, and then we get the distribution of 100 nanometers to 200 nanometers, whatever it is we want is our length of both our viruses and our carbon nanotubes. And we've started looking at developing a technology where we can have phage that actually cross the blood-brain barrier and home into human glioma tumors, which we're now trying to look at in the real-time imager as well, to be able to find small brain tumors as well as study the response of medication to brain tumors. So as usual at MIT, we're never very satisfied, and so I asked my students to do better. I asked them, mice and rats are very small. I asked them to build both a human scale instrument, which we're working on with MIT Lincoln Labs, but I also asked them to build one that was, we could get 3D anatomical information for location and depth without a true scanner. I wanted high resolution from the tissue down to the cellular level. I wanted depth, real-time imaging in living systems, and I wanted it to be inexpensive, minimally invasive, and we'd be able to track a single cancer cell, immune cell, or infection agent in the body. And so they worked very hard on that, and we developed a new system that's called a combined hyperspectral and diffuse near-eye imaging system that we're working on in rats now, where we can see all the way through a rat to 100 micron size particles. So we're at collections of cells right now, and we can see 9 centimeters in depth through phantom tissue, for phantom breast tissue, as well as like meat slices and things like that. We don't have an animal model. The mice and the rats are too thin and too transparent for us, so we need to move on to much larger animal models for that. So that's the end of how you get from beginning of Earth to solar cells, batteries, fuel cells, and cancer. And I'd be happy to take a few questions.
Did somebody say convergence?
Yeah, that was my plan. I saw convergence, and I said, okay, I'll do that.
So on the flip side of cancer is like tissue regeneration. I know like carbon nanotubes have been investigated quite a bit in especially spinal cord regeneration and stuff. Do you have plans to look at that and how you can use maybe some of these materials as sort of scaffolds or for axon guidance or?
We haven't done that, and I have a former student who's faculty at Berkeley. Professor Lee, he's in biological engineering. He's been using it, looking at not carbon nanotubes but phage as guidance systems for cells and in nerves too. So he might be looking at that. It's something we haven't looked at so far in my group, but we're making these really cool, super cool aerogels, like I showed you for those lithium oxygen batteries, and so. That material we're making is both organic and inorganic, and can be very conductive as well. So it might be a good candidate for that if you have a specific application you want to talk to me about. Okay. Yeah. Okay. Thanks for the question. We can't work on everything, but we try.